Good evening, viewer. Welcome to our FTV English words and here's a breaking news. Yeah, from low carb ice creams and keto friendly protein snacks to sugar free sodas, erythritol is a widely used sweetener found in countless diet and specialty food items. However, new findings from the University of Colorado Boulder suggest that this commonly used sugar substitute may carry unexpected health risks. According to the research, erythritol can affect brain cells in ways that may increase the likelihood of stroke too. The study was published in the Journal of Applied Physiology. Our study adheres to the evidence suggesting that a non-nutritive sweeteners that have generally been purported to be safe may not come without negative health consequences, said senior author Christopher D'Souza, professor of integrative physiology and director of the integrative vascular biology lab. Erythritol, yeah, a sugar alcohol that was first approved by the Food and Drug Administration in 2001, and this is typically made by fermented corn, and it is found in hundreds of products and is popular for several reasons. So it contains nearly no calories, delivers about 80% of the sweetness of regular sugar, and has little effect on blood sugar or insulin levels. These traits make it especially appealing to people focused on weight loss, low carb diets, or blood sugar management. But recent research has begun to shed light on its risks too. One recent study involving 4,000 people in the US and Europe found that men and women with higher circulating levels of erythritol were significantly more likely to have a heart attack or stroke within the next 3 years. This was the first author, Howard Perry, a graduate student and his lab set out to understand what might be driving that increased risks. So researchers in the lab treated human cells that line blood vessels in the brain for 3 hours with about the same amount of erythritol contained in typical sugar-free beverage. They observed that the treated cells were altered in numerous ways. They expressed significantly less nitric oxide, a molecule that relaxes and widens blood vessels, and more endothelin 1, a protein that constricts blood vessels. So, meanwhile, when challenged with a clot forming compound called thrombin, cellular productions of the natural clot busting compound TPA was markedly blunted. So, the erythritol treated cells also produced more reactive oxygen species, that is called aka free radicals. So, metabolic byproducts which can age and damage cells and inflame tissue too. So be aware. BNS exclusive report, FTB English, Delhi, Bharat.